Yeah, why don't we go ahead and get started? Um,
All right.
yeah, our speaker today is Dr. Nagesh Adluro from the Weizmann Center at the University of Wisconsin-Madison, who's a collaborator of mine, and he's going to speak on tractography filtering methods.
Thanks, Jeff. Thanks for the invitation. And yeah, let's get started. So we use this quite a bit in our, I would say, day-to-day work. Because of this presentation, I also got an opportunity to kind of go quite deep under the hood with the papers and the tools under the hood a little bit. So this was a good opportunity for me as well. So let's see if I can advance to the next slide. Oh, yeah. So broadly speaking, tractography filtering. So tractography is this streamline algorithms. I mean, it's a vast field, but tractography filtering in particular aims to reduce false positives. So generally, when you run a tractography algorithm off the shelf, like they give reasonable looking tracks, but the more precise questions you ask about specific pathways, specific region-to-region connections, you start to see weird loopy tracks that go off, non-biologically plausible, biologically implausible pathways. And then this whole field of tractography filtering aims to reduce such reconstructions of the fibers. Just to contrast that with other advances that are needed in tractography, like to actually improve true positives and false negatives, that's a different and quite complementary effort. And tractography filtering does not address this part. You to improve, you might still miss things, and you need a lot of, either a lot of brute force computation, better orientation estimation, including novel diffusion encoding, for example, multi-dimensional diffusion encoding, etc., and like streamline algorithms. Like most of the streamline algorithms are local, meaning you are integrating like in a vector field. You do that n times if you want to have n streamlines, and you could do that integration deterministically, probabilistically. The vector field itself could be probabilistic. Contrast that with global, where you are simultaneously doing n integration simultaneously, because there could be crosstalk between streamlines. But I just wanted to list there because that's a different front of innovation compared to this front of innovation in using tractography in neuroscience. This would be much more important in preclinical planning, and this would be more like in neuroscience. In some sense, you could say that. Like, this is much more critical to not miss anything in a preclinical planning when you use tractography for preclinical applications. In the improving, obviously, these measures improves the quantitative nature of tractography, because tractography has this reputation of being like pretty pictures, producing pretty pictures, but not really like giving meaningful biological measures. But improving these things is toward, makes progress towards improving quantitative nature of the tractography outputs and its derivatives, like, extracting specific pathways like uncinate fasciculus, cingulum, corpus callosum, or arcuate fasciculus. And then also with Jeff and I work, with, reconstructing structural connectivities between pairwise regions. You have to get all sorts of tractography connections between different nodes, cortical, subcortical regions. So improving these four measures is towards making that quantitative. This is also, this is complementary to using tractography to derive masks for other voxel to voxel quantitative measures. So this quantitation that I'm talking about in the third bullet is a more intrinsic way of trying to make tractography quantitative, as opposed to the other angle of making tractography, sort of using tractography for quantitative, deriving quantitative measures, is to use tractography to just classify voxels as masks, and then you rely on other quantitative diffusion models like NADI, DKI, I don't know, all sorts of things to GFA, those types of measures. That's complementary. It's you'll see that both are sort of related at the end, but that I just wanted to draw that subtle distinction. And I forgot to say this at the introduction. Please interrupt. And I can't see all the faces, so or the chat. So if there are questions, Jeff, feel free to just interrupt, and anyone else also just interrupt and ask me questions.
Sure.
We'll be more than
Nagesh,
glad.
I have one kind of
Yeah,
like additional comment question. And maybe
yeah.
this is something you'll talk about, but in addition to kind of like straight up say false positives where a streamlined trajectory is just clearly implausible, I feel like there's sort of an issue with double counting streamlines or parts of streamlines as like being part of two fibers with separate sets of endpoints.
Yeah, that's actually towards, I actually have a slide from the paper that talks about precisely why, whole brain tractography is necessary to avoid such double counting and double assignments, how to make, if you have overlapping regions, a track could be connecting uh, both pairs, and then how do you account for normalizing that? Yeah, yes, that's a very good point. You're way ahead. That's almost towards the end of my
Okay.
presentation, but good observation. Yeah, yeah, please. Actually, this is what makes it fun for me also to, I'm also going deeper based on the talk, and yeah, so thank you. Before we dive into the actual just filtering algorithms that we'll go over in this talk, the paper Jeff sent around, I just wanted to say this anecdote, so quantifying tractography is very challenging. So there was this paper published in 2019, fiber length profiling, where they generate cortical tract length maps, which then used to correlate with functional data and other myelin maps, etc. And then after it was published, then there was 3 years later, they published a case study on that paper saying how their data, just a simple thing about calculating length was erroneous. The idea still stands, the framework still stands, but the quantitative nature of it could be so affected, quite a bit affected by simple things that you would assume are correct based on length calculation. This paper I would recommend. I mean, these are good papers. So what they go in that is when people typically talk about a length calculation, there is a step size. There's a very standard parameter in streamline integration, which is you have a step size of how much you want to shoot before you connect the next vector. And if you just use it as a step size, length is counted as step size, number of step sizes. It might work in certain cases. It might not work in certain warped cases where the streamline is warped. And then sometimes you might just do. So don't rely on step sizes was the main point, and it was hidden from them. And what this paper articulates is open software and open forums and discussions. That's where they unearthed this miscalculation. I just wanted to point out that quantifying tractography details matter a lot. And then they just show this is the example of old data when these are the cortical tract maps I was talking about. So they show on each vertex like the distribution of the tract length maps. So like if you see a heat map here, that means this portion of the cortex has a lot of short fibers. This portion has a lot of long fibers. And then they use these maps to correlate with the visual networks, the functional network maps that you would get from resting state. Just to note that the length calculation can affect the findings that you do with these correlations. Just a quick note, I thought I should mention that. So with that introduction and a small anecdote, this methods. So all of these methods are centered around MRTrix ecosystem. MRTrix Donald Tournier, Robert Smith have been using MRTrix for like, I don't know, 13, 14 years. It's very popular, very thorough, detail-oriented, which is needed for tractography. I just wanted to highlight, they just recently got this September 3rd, they got Australia's Premier Science Award for, discovery, innovation, communication leaders, because they really do, they are one of the most popularizers and of using tractography in neuroscience and even preclinical surgery type of planning. I just wanted to, so methods are primarily centered around this paper. It's very nice so this paper summarizes a sequence of developments over several years, but it's a kind of nice summary, everything in one paper sort of thing. So these authors, Robert Smith, David Raffelt, and Tournier, these are all the same team that developed MRTrix. So the filtering algorithms that MRTrix offers are centered fundamentally around this notion of apparent fiber density that David Raffelt published in 2012, a long time ago now. The idea there is that, so whenever you see these tubular-like things, that means they're real fibers, not tracts, not streamlines. They actually have volumes. They're not lines. Lines don't have volumes. So if these are real, not real, models of the fibers. If you have a collection of these fibers, the response function, this is just a cross-section showing the top view. The response function that you get, the signal that you get from such a configuration is this length, this width is proportional to the volume of these axons. That under certain diffusion encoding, which most typical, you need a certain, you can't have too small diffusion encoding times. You need certain encoding times so that diffusion would actually start being proportional to the volume. Imagine like you pour a drop of an ink on a paper, the further it spreads, the more volume it has to spread for. So you need a certain lower bound on the diffusion encoding time, which usually is met in almost all the diffusion protocols. And then this length is proportional to the intraaxonal volume. And then in the downstream protocol, this lobe amplitude is what is the basis for all of the quantitative tractography, or quantifying tractography down the stream. I just wanted to mention that, and feel free to read these papers in more detail. But so the idea with using these amplitudes as quantitative measure, so that's the microscopic group difference. So there is a intravoxel quantification, and you want to extend that to intervoxel quantification. So the challenge in tractography beyond quantifying diffusion imaging measures, that's a whole front we are not even touching, like how quantitative NADI is, how quantitative AFD is, apparent fiber densities. That still is within voxel quantification. The challenge with quantifying tractography is you have to quantify across voxels in a careful way. If, for example, if you're just focused on, if a pathology affects only certain volume in certain dimensions, so these green lines are tracts going up and down, and these red streamlines are going left and right. Then if a pathology has reduction in volume in one dimension, in a certain fiber bundle population, then you would see a shrinkage in these green lobes, because you can see like it's more dense, more volume, more fiber volume, less fiber volume, you can easily detect it. But when you're trying to do, when there is a spatial extent of the change due to pathology, that means that, let's say there is a atrophy that certain voxels are no longer part of the white matter or that particular bundle, that means this is an example of atrophy. But then to compare spatial extent, you almost by definition need a way to get into the same space. So you would find a middle ground with 2 by 2, and then you would have to do warping and modulation and stuff. And if you do proper modulation, then you'll be able to detect even when there is a spatial extent with atrophy that you have a shift, you have this affected, the pathology. You can detect the pathology effect of reduced volume in that. So the entire basis for the filtering presented in MR-Trix is this apparent fiber density notion, this length. That's the key. This is not up. And another important concept. So apparent fiber density is one thing we have to keep in our working memory. And then the pixel. So the notion of pixel, we all know pixel, voxel. So they introduced a term in this paper back in 2015. They coined the term fiber population within a single voxel, pixel. What that is, it's a very simple idea, but very powerful. So we all know the p-value maps when we do voxel-wise analysis. But they say that in each voxel, you can have more than one fiber population. Like, for example, in this voxel right here, I'm pointing to, you have 2 fiber populations. You might have more in reality, but these are the ones we can measure and model. And your effect, the disease effect, even though it's in that voxel, in that region, but it might be only affecting certain oriented fiber populations, not the others, not all of the fiber populations. So you have a significant effect in one direction, but not the other. So this is another idea we have to keep in mind when quantifying tractography. Well, with this framework, yeah, pixel is, not all filtering methods need this idea, but this MR-Trix uses this idea quite a bit. So that's a pixel. Oops. I'm not sure why it keeps not letting me. Okay. The other concepts you would need to keep in mind. So AFD, apparent fiber density, pixel. Then you have fiber density and cross section. So let's go through that. What does that mean? So if you're studying gray matter volumetric differences, let's say there is atrophy, patient lost a certain portion of the brain, you would find that like, oh, okay, it's a smaller volume and that's it. Whereas in white matter, you could have 2 different scenarios. You have a smaller volume, so local difference volume along the fiber length. You have volume reduction in the white matter, but not the number of axons. This is exactly like telling you why the idea of a pixel is helpful. And then if you have such a reduction in volume along, where the voxels are oriented this way, and then you not only have volume reduction, you have a reduction in the cross section also. Your bundle is smaller. Like the entire cross section, if I slice it like this, you can see it's half the cross section. So it's not only the density that is smaller, but also if you imagine cutting through vertically, perpendicular to this, here it's like bigger cross section. Here it's like half the cross sections, and also lower density. So that's the pixel concept allows you to model these things very effectively and capture those subtle effects. So like here, each gray dot is like an axon. These are, cartoons, but just to give you an idea, like you can have a situation with a disease where you have reduced fiber density. You can see like visually, literally. But the cross section is pretty much the same, because if you kind of slice through, then yeah, you have the same cross section macroscopically for these 2 scenarios. And then you can have the situation where cross section is smaller, but the density is the same. Like there's loss in somehow like it got trimmed around the edges, but then there's an intact portion inside or something, something like that. And then you have, I should also say while I'm talking, these notions of fiber density, fiber cross sections, these are still interpretations. So keep that in mind. I mean, these are plausible interpretations of these measures, the amplitude, basically using that. But there's always a fuzzy bit of link. The link between these amplitudes and true biology is always a little bit fuzzy. Um,
Nagesh, can I make
yes, yes,
a comment here? I mean,
yeah, yeah,
I think this would be sort of an interesting point to think about,
yeah,
like what sorts of these changes we would hypothesize, in different like neurodegenerative conditions or other sorts of like neurological insults. I mean, um, my I think like what in the case of significant like global white and gray matter brain atrophy with like expansion of the ventricles, I would think perhaps either the bottom right case is the reduced cross section with or without reduced fiber density because you've got kind of um, expansion from the ventricles that's like compressing like white
right.
matter that's degenerated. But um, uh, I don't think that's maybe the only scenario here, and I'm curious what other folks would hypothesize. Like for example, in with like a primary white matter disease without a lot of, say, global atrophy, what would you
Mhm.
expect?
That's the CS, the expansion of ventricles is a great example. Yeah, I agree that that would cause more of these two types of situations. You're asking what would cause sort of this where cross section is intact. Maybe loss of myelin is one example. I'm not sure. Like,
Hmm.
because they say the cross section is a proxy for information capacity. So you can imagine these fibers losing the volume could be lost by that sheath, that wraps around these axons. Maybe loss of myelin would be this scenario. That's again, I will admit my limitation. So I have to read and hypothesize more on what kind of insults and neurodegeneration might cause specific types of, these things. But that's something I can look in literature and maybe, yeah, yeah,
Yeah, I wonder if like the people who created the SIFT algorithm proposed
yeah.
different sort of profiles corresponding to different clinical scenarios.
I think they did. I will admit, I that's not hashed in my working, set of memory. But I will. What I can do is I can try to look into those papers and make a list and like summary of what this team in particular explored, and or the teams which explored this in particular did. Most of the times we also use this, but we left it at the point of epilepsy. So I mean, towards the end you'll see some examples of publications. We use this method in Alzheimer's disease, but we usually stop at uh, saying the cross-sectional area is different. We haven't done like fiber density versus cross-sectional area, that like kind of, hey, the cross-sectional area is different, but we didn't check whether the fiber density was different also. You'll see why, because the filtering actually produces the cross-sectional area, not the density. We could have run models where in epilepsy and AD, okay, fiber cross-sectional area is reduced, but what about fiber density? We haven't run those analyses just because the SIFT and these things don't produce those readily. But based on your question, we should probably run those models ourselves. But I look for if others have run that, and then I can report back to the group, to you and the group. That's a very good question, very good point, because from a neurology and neuroscience point of view, those would really make get a lot of importance. Okay. Every time I have to click here. Okay. So I briefly mentioned the quantification within and then across voxel quantification. This is a busy figure, but basically, let's say you have 3 nodes on the left and 3 nodes on the right. The entire set of streamlines is a whole brain tractography. And when we talk about an edge connecting 2 nodes, these are the dark black lines. Those are the bundles that belong to that edge, or the bundle. You can and you have from A through D. These are showing microscopic voxel level quantification. So the fiber density part comes in. Like you can have a voxel where you have low density, and here you have even lower density. You should think of, so voxel can have a high density, but then the intersecting part will have a low fiber density, and you can have really high fiber density and then half. Like you see, each voxel can have a certain fiber density, but the fiber density attributed to the bundle can change voxel to voxel. What this tract across voxel quantification aims to give you are the E and F. Let me tell you what that is. So E and F, even though the length here is different, they are identical. Like what that is you're getting the cross section 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So you're summing up the fiber bundle cross section across the five streamlines. And that's called macroscopic. So the cross section that we are talking about is not really what you're seeing here as this visual. It's the sum of these little perpendicular red hotspot, like vertical lines that are so the cross section is invariant. So the point here is that when you are trying to quantify across voxels, that's what you're trying to do with the bundle. You want to have a quantity that is not changing like as you move along the track. So that because you want to summarize that tract in a quantitative way. That's what this figure tells you, is that at the end of the day, the fiber bundle capacity, that's an interpretation. So there's links of interpretations here. So the intra-axonal cross-sectional area that you get, they say that is actually proportional to the capacity of conducting information between the nodes. That's interpretation, but yeah, but that's what you get. And Jeff, I think when we ran the analysis in AD and epilepsy, we get this measure. We get the fiber bundle capacity. That doesn't directly get at your question of fiber density. For that, I think we have to run actually pixel-based analysis, which is much more computationally intense, where we not only run the bundle capacity, but also like some sort of density changes for the bundle, which we haven't run. And that would tease apart whether it's a reduction in density only or the reduction in cross-section area. So that's a good suggestion. Yeah. So in the past, pixel-based analysis has been super duper computationally intense, forbiddingly so. Like our group tried to be one of the early adapters of the pixel-based analysis, and we just couldn't, the resources we had weren't sufficient. This was like, I don't know, at least 5 years ago. We haven't tried pixel-based analysis recently, but that would answer those questions. Okay. Okay, now we are actually getting into, so that was like still sort of introduction, I guess. I know I put in methods, but now we're actually getting into how this is accomplished slowly. So this is the overview. So SIFT stands for like spherical deconvolution informed filtering of tracts or tractograms. So what we are seeing here is that, so this is the overall flow. So let's say you have this like true biological white matter bundle. Like this is the actual object we are trying to image. And then, zoomed in, if you zoom in, you'll have the density, which is like 1 million axons per a volume, like millimeter cubed. That's the truth. And diffusion MRI tries to get a approximate picture of that, use a diffusion model, and then you get the fiber orientation. Remember, this amplitude is what we are gonna try and take advantage of as a proxy for quantification. But then you use this FODs to then do the streamlines. So these are the reconstructed streamlines of this fiber bundle, the biological bundle. In reality, we can typically do on the order of, like, computationally speaking, we could do about 1,000 streamlines per millimeter cube, or maybe 5,000-10,000 at most. I mean, there's a paper called Blue Tract, I don't know if people know, where they actually generated 1 billion tracts in the brain, and then, I don't know, they did it, but that's just not feasible on many empirical studies, in many labs. But typically, it's 1,000 factor difference between the in the density, in the volume that you're trying to get. So the key is to how do you go from these streamlines to make it proportional to reflect the fiber volume. And the key is to take advantage of this globally, these amplitudes, and try to match. You reconstruct FODs from streamlines, you reconstruct FODs from images, you try to improve the consistency between that, and then say, okay, this is the best fiber, apparent fiber density I would get. So that's the idea. So like you take the fiber densities, you take the streamlines, they call it semi-global because the streamline is not global, but then the calibration is going to be global. You'll see the streamline is still local, but the adjustment of the weights of the streamlines is global. You take the whole brain tractograms. That's why they call them semi-global algorithm, but that's just semantics. And then you would derive the fiber bundle capacity, which you hope, that's why they put it as a dot. These guys are super detail oriented. You'll see, notice that in their work, in their presentations. This is a proxy to that for this. It's not real connection. So, okay, that's the overview. Again, fiber bundle capacity here. This is we saw this before, but essentially like the fiber bundle capacity is sum of all the bundles they contribute to the area. So if two nodes have lots and lots of fiber bundles that contribute to the lots and lots of volume, apparent fiber density, there's a lot of fiber density, and then you get a cross section, that connection is strong, or there's a lot of capacity in that fiber bundle. That's the idea. So this is the example bundle connection that you get using Tractoconnectome between two regions. Yeah, different views, and you see how there are all these tracks that might or might not exist, these loopy tracks. The idea is to downweigh these and these extraneous loops that are like solar flares that are coming out of the and then focus more on the real connections. No, not real, I mean, yeah, the actual connections between the two, and then so we'll go through like not in super detail, but at least overview of 4 algorithms that try to get at that. And good thing is they're all packaged. I mean, to actually get the streamlines, you could just use Tractoconnectome. To get the algorithm one, there's always accompanying MRTrix tools that go with these algorithms. And the idea is we'll go through these. Each gives you sort of, this is the loosest, which is giving weights to all those loopy tracks. These are max MIPs, max intensity projections across the brain in the coronal dimension. But they claim that this is the most reasonable looking. This you're giving too much weight to all these loopy tracks. The idea is that this would represent, so this D would represent a biologically most plausible connection between the left and right regions. This is just one example, but I will also say that the quality of this filtering is not uniform across all pairs of regions. It might matter because your lower bound, I mean, your sort of upper bound, I guess, of performance is limited by the streamline algorithms to begin with, which I said is a different front of innovation. So your filtering can be only as good as the underlying proposal distribution, so to speak. I don't know if you know particle filtering. So if you shouldn't expect when you do in practice that all pairs of gray matter cortical, subcortical regions would behave so nicely. I mean, it depends on a lot. It's very coupled with the underlying streamline algorithms as well. So, okay, without further ado, I guess we could go into algorithm 1, unless Jeff or anybody else has questions. But okay, so what does algorithm 1 do? So these are, so you have region A. I don't know if you can see still on your screen, there's a little color difference here. This is node A and this is node B. They're colored same. And you have these streamlines that are assigned to that prefrontal. I mean, these are cartoon representations of the prefrontal bundle. And these are the streamlines that are connecting region A to region B. What the first, let's see the simple formula. It's very straightforward, actually. I mean, not to implement, but to understand. I mean, once somebody packaged it. What this is doing is this length, as I said, is proportional to the fiber density. That's why I put it here up here. So always remember that the quantitation is coming inherently from this. So whatever the limitations of this are, that will carry through. But assuming this is reasonably quantitative, you would just sum. So you take all the streamlines and see the intersecting pixels. Remember fiber populations, like, so these would intersect more with the, like, for example, you can see that this black line is, so red is intersecting. The fiber population that's going this way is not intersecting. So you would have to have, so there's a notion of definition of intersection, and then you just sum up the fiber densities of all the pixels, very natural. It's almost like using the map. And then you normalize by the average stream length. This is just the streamline count, which with this new denominator is basically the average stream length of the bundle. Like, so it's like fiber density divided by the average streamline length. So that's the algorithm one, which gives you this kind of a map. And then from here on, it just becomes a simple improvements over, here it's more binarized, right? You say whether a tract is intersecting a voxel or not. Here you relax that and just use the streamline density fraction, like how many of the streamlines of the overall. This is where the semi-global thing starts to kick in. You're now using the overall whole brain tracts, the dotted lines, and you're saying what's the streamline density fraction that belongs to this bundle as a fraction of the whole brain tracts. And then you're no longer then restricted to the intersecting pixels. You just, because this is a soft weighting of the fiber density instead of like literally binarized. So this is a soft weighting of that, and denominator stays the same. I mean, they list this because this just shows each algorithm has limitations and pros and cons, but shows how a unifying way of thinking about deriving this. Technically, it's a different kind of FBC. The formula is the right-hand side is different, so this must be like FBC2 or something, but yeah. And then this is the algorithm 3, which now decouple, like instead of using the average streamline only in the bundle, you're going to now say, I'm going to use all of the streamlines. So you're slowly making it into more and more global. Like first, we were only using intersecting voxels and the bundles, streamlines within the bundle. You relax the numerator, now you relax the denominator, where you're just going to say, okay, all the streamline lengths and all the, I'm just going to, but then I'm going to just sum the weights at the end that belong to the bundle. So you're not bringing the bundle when you're doing the ratio. You're just saying, okay, I'm going to do it for all this. The weights will be for every streamline. And then for a particular bundle, I'm just going to sum up the weights. Then this is the main one that we use for all our work, is, so instead of like doing this closed form solution, like for the weights, they actually have like an iterative method to get the weights. And that I mean, at some point we can go into it, or maybe another time, but that's a really deep, you have to use reference image, and you have to have the, matching between the streamline density from this and and then you do iteratively optimize. The main challenge, the main reason they did from here to here, is that, so this would work. Actually, this is a really good formula. From here to here is because of computational restrictions. I want to point that out. If you had immense computational power, you would generate lots and lots of tracks. Like, let's say, I'm just exaggerating here, like, I don't know, 100 million, 200 million tracks. This would actually work better. But the problem is, in practice, we are not able to generate barely, we are barely able to generate 20 million, 30 million if you have a 100 subject study, space limitations, computational limitations. this tries to overcome that practical limitation by not needing to generate lots and lots to filter from. Rather, you're cleverly adjusting weights so that you're bringing creativity in weight calculation, so that's just a qualitative statement, but the details are in the SIFT2 paper of this particular optimization.
Nagesh, can I
Yes,
ask,
yes, yeah,
so
please.
like the reference image for the SIFT2 algorithm, does that have to be, is that ideally from your own data set or acquired with the same diffusion imaging protocol, or is that
Yeah,
some kind of like more,
gold
I don't know,
standard.
more general yeah, average template or what?
Good question. No, this is all within subject still. So this is still within subject. You're using the microscopic fiber density as opposed to streamline density. So this is all of these are all of these are based on individual subjects' own data. When they say reference image, it's more like there's one figure I didn't include in the presentation. It's like its own fiber density image, sort of like taking that into account. You're iteratively matching. So you might ask, well, that's the case even in this picture. Yes, you are using the fiber density. This is still you're using the not here, here. Even here, you're using the same fiber density of the reference image, which is its own FOD image or apparent fiber density image. But the key thing is here you are filtering, because this itself is a filter. If you see what I mean, you're dropping lots and lots of. So you're calculating with this for 100 million, not 100, 20 million tracks. And then when you're actually getting fiber bundle capacity, you're summing only, I don't know, at most like 1,000, if that, depending on the pair. So you're filtering out all of the tracks that don't belong to bundle right there. Whereas in this case, you are using all of the tracks to optimize the weights, even though you're summing the bundle weights. So this bundle weights, these WSs have seen other streamlines too. Like it's a very coupled process. Here, this is per streamline only. Whereas in this case, this WS has seen information from the whole brain tracks. That's a long answer, but the to the point answer is this reference image is its own subject's FOD image, basically, the apparent fiber density image. The native, there's no external information that's brought into this algorithm. It's the same diffusion image that's used in this process. It's just that these weights are more global, sort of. You're adjusting the weights based on what you see in the rest of the brain and iterating it over and over. Yeah, so that's the idea. And algorithm 3 and 4 are very closely related, actually. That's why I said it's a computational tweak as opposed to real conceptual tweak. I don't know if you know what I mean. Like, it's a computational tweak as opposed to a conceptual tweak between algorithm 3 and 4. Okay. All right. So then what are the FBC problems? What do people can take away from this FBC, right? Like, you're getting all this. This is by design. It's not like emergent properties or anything. So it's like it scales with the cross-sectional area of the bundle. The bigger the cross-sectional area, the FBC will be bigger, and scales with the fiber density, but in a way not. Yeah. So if there is a bigger fiber density, you would get bigger FBC. But to your earlier question, Jeff, like whether can you capture because if cross-sectional area increases and then fiber density goes down, FBC might not change, right? And you won't be able to detect that. Like if these two go in opposite directions, because they both scale with fiber density, but if both are opposite effects, then FBC is going to be blind to that effect because they cancel that effect, which is why we have to do a separate FD analysis. So that's a limitation in some sense. That was brought to light by your question. People who have been doing tractography for a while, they always say there's length bias, and this is invariant to the length of the bundle. Like when I showed you that cartoon, it's across voxel quantification, it stays uniform. It's across voxel quantification, doesn't change by the length. And also it's invariant to the number of streamlines used, except the number of streamlines indirectly matters if you don't have enough to begin with because think about this, if you do not have enough streamlines to begin with, then this set becomes sparse. Then it's sort of the stretched claim that it doesn't matter, but actually it can matter practically for certain regions if you're not representing the all the eigen tracts connecting two pairs. Because if it's one thing, you have all representative exemplar tracts between the two regions. Let's say there's only five and five only exemplar tracts, then it doesn't matter as long as you get those five representative tracts, 5 or 50 or 500 or that. But then if the number of streamlines is so low that you're not able to capture the representative tracts, then it does matter. But that's by design. And there are some discussion points. As I said at the beginning, this could be complementary to what a lot of people do. Jeff and I also did. Jeff uses GFA. You can actually extend that kind of average summary, but you can, there is algorithm 5A and 5B, which I didn't present in the paper, that you can, so when you compute these weights, so we are relying on streamline density fraction. You can actually use other measures that you care about. I don't know, they could be some NADI, GFA, or other things. You can, but those require like implementing things. And so you can bring in per pixel, per fiber density, per pixel quantification, even if you want to just say, oh, I just want to know what neurite density is across for this bundle. You can actually optimize weights per pixel based on other metrics as well. Some of the other things that people generally think about is, oh, what about bigger brains and smaller brains? They actually lean into that criticism and basically say, like, FBC, yes, FBC may be proportional to the brain volume, but it may not be an unwanted confound. I mean, because they claim FBC is a proxy for information capacity. If somebody has a bigger brain, or which is proportional, if that particular bundle, again, the relation should be based on if there is a bigger corpus callosum, there might actually be a, that's what they say at the end. It's a discussion point. These are not, and then whole brain tractography helps. I have a picture for what Jeff asked at the very beginning. And then obviously this is not the only player in town. I mean, you have Comet, Life, all of these are similar in spirit. They enforce some notion of consistency between the reference image or the imaging data and the streamline data. It's like imaging and streamline, how do you make them consistent to calibrate the streamlines, basically. So this is what Jeff was referring to. I think whole brain tractography helps because they kind of make a push because when I was trying to do these connectivity maps, I was thinking maybe I should just do pairwise tractography and maybe that's going to be better and stuff like that. But they actually say for better comparison between, for better calibration between different bundles, it's better to do whole brain tractography than pairwise tractography. Because let's say you have these two gray matter regions, this is the true fibers, and then using your modeling, diffusion modeling, apparent fiber density, you get these pixels. Remember, pixel is here we only have one fiber population, right? If you had like a other direction. So here it's pixel, and the thickness actually indicates the density of the fibers. That's the proxy. This is the measured and modeling process of this underlying this thing. And bundle one, so when you're trying to assign tracks between green and green, so obviously this assignment, this particular stream, pixel set belongs to both the edges. And when you are trying to, if you did not take into account the whole brain tracts, remember this WS takes into whole brain tracts, like it's taking all of the tracts into account when calibrating these weights. If you did not do that, if you only fed, let's say, only a top bundle or a bottom bundle, then you would miss that calibration that spans these two adjacent nodes. So that by making the semi-global calibration, you're with the whole brain tractography, you're accounting for that double counting, or I don't know. Is that what you said, or did you have another complementary question about this, Jeff? I mean, does that roughly get to your double counting question, or?
Yeah, I think so. Yeah.
Okay, okay. This is just data I wanted to show because this is not the only player in town, and there are other methods like, well, SIFT is, you can say these two are similar, algorithm 3, algorithm 4. This is different, and commit 1, commit 2, you could say they're the same. Their performance varies quite a bit between binary, if you binarize the graphs, versus weighted. And weighted is all like that washes out a lot of the differences between the filtering methods. The motivation is all there for these methods. So these are based on numerical phantoms. This is the other paper that Jeff forwarded. I didn't want to go in too much in depth on how they did it, but I just wanted to point out that these methods are great, but they're not always like standing out as a winner or something like that. That was my main point. And then for spherical numerical phantom, this is much more. So this was based on some sort of a HTP model. So they took like 10 subsets. They got the diffusion models, and then they simulated back the diffusion signal from those, like a human, to mimic sort of a human brain phantom. Whereas this one is purely numerical, and then the differences are even smaller if you have a very controlled, it's almost like a sanity check between the different methods, because many times methods are compared in suboptimal settings, like you don't give the method the best possible use. So it's like sometimes one method uses the other methods in straw settings. And I just wanted to mention that, like, filtering is very challenging. But the main takeaways are that, like, from that comparison paper, at least for simple tubular fiber bundles, like I said, it's not the same for all region to region connections or all bundles. It depends heavily on the geometry of the bundles, underlying biological, geometry of biology. For simple tubular type bundles, where you don't have fanning or kissing, or these do improve. Filtering algorithms do help. For more complex brain fiber architectures, they're going like super U-shaped, or like short fibers and fanning fibers, the improvement is modest if you're being generous. And then obviously, filtering algorithms have the potential to improve connectome mapping, and particularly for probabilistic and weighted, and then innovation is obviously needed. We do use it quite a bit. Like I said, we have all these papers that we published. We did not ask the question that we did not pursue the question Jeff asked, but hopefully we will in the future. The top is only abstracts, though. These are not papers, but we did like use the same fiber bundle capacity, and then derived graph theoretic measures. We used edgewise analysis. You could do graph theory analysis. You could do edgewise analysis. We did all sorts of things with this. And yeah, sorry. I don't know. That's what I have. And happy to take more questions. I see something in the chat. What is it? Oh, can I click that? I don't know.
I think Sandy was just thanking you and saying he has to leave for another meeting.
Oh, yeah. Thanks. Yeah. Yeah, that's all I have for today. Thank you. Thank you, Jeff. And I don't know who else is. Yeah, everyone.
Yeah, no, that was super comprehensive. Does anyone have any questions for Nagesh?
I have a quick question. Oh, thanks for the great presentation, by the way. So you mentioned, like, you showed a bunch of examples of, even within that one paper, of different algorithms, and then there's other algorithms. I guess from like a practical point of view, how do you know, like, what is the best for your data or for your problem? Like,
yeah, that question depends on the answer to that question depends on how much a priori knowledge you are bringing into the game. Like the best is dependent on, hey, if you come in with a hypothesis that I think that the effect is mainly in the tubular bundles, or the effect is mainly in the short bundles, then you can start, the more a priori knowledge you bring in, the more you can distinguish these methods to particular type of hypothesis. But there's no universal answer. Like, yeah, if you have a uniform prior from, you don't know where the effect is, stuff like that, almost all the methods are statistically going to be equivalent. Yeah, that's my understanding, and I've been doing it. I mean, absolutely. And I will tell, like, if you have specific bundles in your hypothesis, like uncinate fasciculus, where you can specifically isolate them from the streamline models up front carefully, you know, that's where that's the best case scenario. And then there, I think it also depends on whether you want to have a fixel. So commit and life, these are not, so in fact, these are not mutually exclusive even. You can combine SIFT and commit. So like SIFT is fixel specific. So commit is just like focusing on, I'm going to get like more relevant streamlines, and it stops at that. It actually doesn't go from relevant streamlines to fiber bundle capacity or fiber density or cross section. You can combine, you can use commit, and you can get SIFT based quantification of fiber density, fiber cross section. So it's not like one method is, you can combine the methods. So that's my answer. Sorry, hopefully that helps a little bit. Yeah.
No, that helps a lot. I guess just from like a very naive, I'm not very familiar with this. If I was interested in using this, like how do I know that I got the right answer, kind of thing? Like I'm happy to like try a bunch of these, but then I assume that they're all going to give me slightly different answers. And what is like a good approach to be like, oh, this looks like this is the right method, kind of thing.
Usually, like, so I could give you a practical answer of how we publish these. So practically what we did was, we started just with SIFT, and we ran like graph theoretic. You could derive weighted graph-based graph theory measures. We did edgewise analysis, and then like ran those statistical group difference. Sorry, I didn't include plots from these papers. So happy to connect with you offline. But then we consult with like neurologists and say, okay, do these make sense? And yes, they do. Yeah, they don't. And it's basically a loop. There's a human in the loop, sort of. You do, we did SIFT, and most of the times they did make sense to the, again, you could say that's a bias, but for the most part, they are interpretable. Let's say by make sense, I always mean they are explainable. They are rationalizable. They make rough sense. It's not like, yeah, we didn't compare the head-on between commit. I mean, SIFT is one thing you can go by for your practical choice, though, is like SIFT is very practical and used a lot for deriving the graph theory measure. So it's, in some sense, there is emerging literature evidence that is piling up to talk about the consistency and of these methods. So SIFT is a fairly safe bet for your practical choice if you wanna use one of these methods. Yeah, it's a safe bet. Yeah.
Okay, thank you.
Thank you. Yeah, I'm gonna stop sharing screen and
Cool.
yeah.
I guess I just have one more question, which is,
Yeah.
whether you think any of these algorithms are sort of more or less sensitive to, say, differences in diffusion, like sequence, like diffusion protocol. Like, are some of them better if you're going to be trying to combine like multi-site data sets, for example, something from like
Excellent
ADNI
question.
or data that were acquired over many years with like sequence changes?
Excellent question. Actually, that's why I kind of highlighted the AFD part. I remember I can share the screen again, but Yeah, why don't I share? Why don't I indulge a little? So actually, yes, that actually is a very good question, because the entire premise of this fiber bundle quantification goes back to this. The reliability of the connection between the length of the FOD and interactional volume.
Mm-hmm.
That actually requires fairly high B value, at least 2500 or more, to have that link between the length of the FOD lobe and the interactional volume. If that link is broken, the entire thing falls apart. Like at least, I mean, you can replace that with other quantitative measures or something, because fixel, the notion of fiber density is no longer true. You can use, for example, I don't know, NDI, or like I said here, you can use complementary quantitative measures, but not fiber density, for example. So the idea is that you would have to replace these internal calculations. Instead of fiber density, you would have to use, let's say you only have BF 1000. The best you can get, the best quantitative measure you can get is mean diffusivity. FA to some extent, maybe FA or MD. So then you would have to go in and the only thing this framework offers is the notion of fixel that is decoupled from the acquisition, but also the quality of what each voxel, fixel is nothing but you have to have a notion of fiber orientation population in each voxel. So if you have only DTI, then you might only get one orientation that is reasonable. So yes, acquisition does matter, because the definition of a fixel, the definition of fiber density, all depend on this, good enough B values and high enough SNR. Yeah.
Okay, interesting. Thanks. That's very helpful.
Yeah. Thank you. Yeah. Thanks for all the good questions.
All right. So maybe we'll wrap up here. Again, thanks so much, Nagesh, and I guess we'll see everyone in 2 weeks.
Yep. Thank you, Jeff. Thanks, everyone. Yep. Bye.